Welcome to each and every one of you. Some of you have traveled from quite afar, as I say at Indian weddings. Those who've come from the biggest distance receive the greatest applause, and there are two sitting right here who are good friends of ours, which I'll talk about a little later. So again, welcome. And as you'll see, this is going to be a conference with a representation from a lot of different entities, if you will. There will be a lot of mixing of ideas, I hope, and that should be the spirit of this meeting. Willingness to think outside of the box is a given. It'll be a given at the Morningside Center, and it should be a given at this conference. So there's no barrier to what you should be thinking and how ambitious we should be. So with that tone, let me take a few minutes and talk about this topic, which has been on my mind, as many of you know, for several years. So the problem we're talking about really is quite straightforward. If there were no unmet needs in medicine, we wouldn't be here. It's quite clear that there remain large unmet needs, and many therapies that we have currently available to us are expensive. Many of the newer ones, in fact, are quite toxic in many cases, and only modestly effective. And so the opportunity here is the following, that it is our belief collectively, and I suspect many of you share that there exist promising ideas that are just not moving forward, or not being developed, meaning either put to a test to be either validated or shown to not work because there is an insufficient financial incentive. And a few years ago, my wife and I, Vidula and I, started an organization called Global Cures. We sort of pinned this name called Financial Orphans to these kinds of ideas. So let's just think about how drugs are developed from the standpoint of a new drug that's being developed. Typically, one has certain preclinical studies to identify a target, identify a pathway that one wants to intervene in. And many of the illustrations I will use will be from the cancer world, since a lot of our attention has been in that direction. But a lot of what I'm going to say today applies outside of cancer as well. So preclinical data, initially perhaps in cell culture, then in animal studies, leading to ideas that sound interesting to move to the clinic. But the key question that arises is whether there's an adequate return on that investment. And that's assessed typically by whether there is a patent, in fact, that has been filed to protect that intellectual property, that idea. Is the market size large enough? What are the chances of successful development? Meaning how well do we know the biology, and do we think that that target is going to make a difference at the end of the day? And reimbursement. Many ideas are not pursued because there is no reimbursement, or may not be a reimbursement. And so if the ROI is felt to be large enough, pharma, biotech typically takes up these ideas. And if that's not the case, these ideas, even for new drugs, will languish. Now, what about existing drugs? Here, we may get promising ideas from two other kinds of evidence. One is epidemiologic evidence. And so is there information in the literature already that suggests that the use of a certain drug or drugs changes either the incidence of a disease or its outcome? And so that is another feeder into this. And occasionally, because these drugs are available, one will hear case reports, sometimes phase 1 studies, sometimes single-arm phase 2 studies, and those sound quite interesting. So all of these, for existing drugs, feed into the pool of promising ideas. Now here the problem is that in many cases there is no IP. Now in some cases there's a new use IP that might be filed by the investigators, but if that's not the case, it's already in the public domain, there's no intellectual property. Even if there is a new IP filed for a new indication, in many cases, at the price point the drug is at, that new market may be very small compared to the original market. So if you take a drug like metformin that has about 50 million prescriptions a year, at the price point of metformin, even if I were to show you data that metformin might help as an adjunct therapy in cancer, there is no incentive really to move that forward, even if there were an IP for the new use. Now, of course, one could reformulate the drug, and that is being done by pharma and biotech in certain cases, either to improve its pharmacokinetic, pharmacodynamic properties, perhaps optimize its mechanism for cancer, for example, which may be different from that for its original indication. Or just because that's just the way to get IP, that is to modify it sufficiently so that IP is available. But if none of these things happen, these ideas for repurposing of these drugs in general will languish. So that's one category. Another major category is nutraceuticals. In certain cases, pure compounds, it could be vitamins, et cetera, but molecularly, purely characterized, well characterized. In other cases, that could be herbal products, and in other words, mixtures. What about lifestyle interventions? If I were to make an argument based on some preclinical data and perhaps some human data that it's not such a good idea to eat sugar in a cancer that is FDG positive, you might say, well, show me the human data. So again, who's going to run those trials? It's not going to be biotech and pharma for obvious reasons. And again, just to be clear, my view and our view in general is that we are not at all against biotech or pharma. This is just not in their appropriate scope. So has this problem been recognized? So this nonprofit I mentioned a few years ago is still alive and well, but it's a small operation. We started about 10 years ago, and 3 of the original folks who founded it are in fact right here in the audience, and we thank them for all the work they've done and continue to do. The Anti-Cancer Fund, I mentioned 2 of our visitors from Brussels. We have been very much engaged with them over the last few years, and this REDO project, Repurposing Drugs in Oncology project, was an outgrowth, in fact, of that effort. Cures Within Reach, the former CEO of that organization is also here, Bruce Bloom, and that's another organization that's really highlighted this issue. Now, the government is slowly, but I hope deliberately, walking into this space, and we will be delighted if they do. And we've appealed many occasions to NCATS, Chris Austin, various groups at the NIH, at the FDA, at CMS. Why CMS is the largest insurer in the United States, of course, and some of these ideas may prevent cancer recurrence, for example, just in that space, and therefore may save money. We had invited several folks from the government. Is there anybody who has come from the NIH or NCATS? I'm curious. There is one person back. The FDA, thank you. And in fact, so one of the nice things that's happened, and they were not able to come, several of the folks we'd invited, but they have actually turned around, and I'm delighted that they're organizing a meeting on December 5th and 6th around repurposing of drugs, and really focusing much more on the regulatory and funding issues, which we'll allude to in just a few minutes, as well as some of the science. And this conference will focus a little more on the science and perhaps a little less on some of the other issues, but they're equally important. In academia, this has really not grabbed a lot of attention. And the Morningside Center, I think, is rather unique, as Dr. Lewin mentioned, in really focusing the attention. And so we came to a conclusion about a year or 2 ago that we needed to play in a larger sandbox, that what we were doing in our nonprofit and global cures was just the tip of the iceberg. We needed to engage in advocacy. We needed to bring in folks who might have smart ideas from the world of law or business to potentially consider changes in how we move such an agenda forward. And of course, we needed help from the funding standpoint as well. And so being part of the university and embedding this university allows us, in fact, access to those resources and not to have duplicate some of the efforts. So we're very excited about that. And we're hoping that there'll be others who will join this effort as well. So the advantages of this kind of work are actually fairly straightforward. Many of the drugs are quite cheap. The toxicity in many cases is well known. In some cases, if they're used in a different indication, it may or may not be, if they're used in a different dosing or different scheduling. And so that's got to be looked at. But typically, the toxicity is well known. And there's wide applicability, all of which translates into saying that this could have rapid and worldwide impact. Now, there are at least 3 challenges, and I'll only point to 3 at the moment. We need to identify these interventions. And so, where are the greatest opportunities? And then how do we prioritize these? So I want to say take a few minutes on that. We need to find investigators who would want to move such studies forward. And of course, we need to think about funding. So a few minutes on each of these topics. Identifying interventions and prioritization. So again, from the world of cancer, let me cite a few examples. And I'd like to say that some of these interventions are non-cancer drugs. And these fall into sort of 2 buckets. Common drugs, so it might be beta blockers, for example, or metformin, as I mentioned to you today, ARBs, and so on. And those ideas for repurposing of those might come, in fact, from epidemiologic studies. There are other drugs that are on the market that are rather what I would call in the uncommon category, and I'll show you some examples of these. We're not going to get those ideas, promising ideas from epidemiologic studies. Those would largely come from preclinical research or in silico work, as we'll hear about a little bit today, or, of course, wet lab work. And these case reports and limited trials will generally be with uncommon drugs, and generally also in the cancer drug direction. The reason I mentioned these for repurposing is that they may be used in other cancers than initially the indication was for, or we may use them in a different scheduling. Low dose use, for example, of certain chemotherapeutic agents as an example. So these are sort of the various different buckets and how they might come about. Some examples, these are just 5 or 6 examples. There literally are 50 to 100, depending on where you put the cutoff on the level of evidence that you want. More than that, sort of common drugs that have been identified that are in the literature that may support their use for cancer treatment. Here's some uncommon ones, and these are less familiar with folks, for example. So fenbendazole is an interesting anti-worm agent used in animals. Veterinary medicine, melatonin, there's an interesting literature on it. Chloroquine and hydroxychloroquine, we'll hear a little later today from one of our speakers about the use of that drug as a drug that blocks the last stage of an autophagic response, and so on and so forth. We can spend a fair amount of time. There's some very interesting drugs here with some very interesting biology. And what about cancer drugs? Again, used in a different way than what they're currently being used. Retinoids, capecitabine, HDAC inhibitors, and so on and so forth. So the problem here is that there are a lot of options. And it's not at all clear how we end up prioritizing these. There's not even a consensus as to how to best treat cancer, for example. So this is why this area is complex. And what I'd like to do in the next few minutes is show you a framework that many of us have been working with sort of behind the scenes, if you will, over the last few years. It's not as if I'm going to say something that's incredibly novel here, but I am going to put it in a way that I think will appeal to many of you, and it's something I think that we should be thinking about. And that is 3 broad ways, all intersecting, to think about the treatment of cancer. That is addressing the adaptive response, addressing the so-called injury response, and thinking about instigating an immune response. So here's the basic concept. In cancer, as you know, there is uncontrolled cell growth. That puts a certain stress, which I've referred to here as cellular stress, on those tumor cells. Now I'm talking about a solid tumor at the moment. How does a cell, what does a cell have to do to live in that, when you really come down to it, incredibly harsh environment? How does it get oxygenated adequately? How does it get the appropriate nutrients? How does it get rid of proteotoxic stress? That is, we know there are lots of mutations in cancer cells. Those mutations generate proteins that are mutated. A cancer cell has to dispose of those. So it is under that stress. And there are others that I'll come back to. If those stresses are adequately addressed, as shown up here, then there is cell survival. If they're not, there's cell death. But even in death, a cell can be dangerous. And I think more of us recognize that. It's not as talked about as perhaps should be, because the body has a stereotypic injury response, in fact, to the cell death that undermines the fact that a few cells have died. In fact, helps to regenerate any population of cells that may not have died as a result of that initial stress. So when you add these two together, that is that there are factors now as part of the injury response that are secreted more in just a couple of minutes. In general, the injury response, unless there is a non-sterile aspect to this, if there's a sterility, in general, that response is immunosuppressive. There's a fibrotic response that can ensue also as part of the injury response, all of which feeds into tumor growth and metastases. So that's at baseline. That's a tumor that's expanding. Now, if I give a cancer treatment, the same holds. And so these responses, essentially on the left, for example, are undermining the very initial cancer treatment. And there's data to show that if we accompany cancer treatment with ways of blocking that injury response, the outcomes are improved. So here's the injury response in a little more detail, hemostasis. Then the question is there sterility or not? If there's not, there's generally immunosuppression characterized by cells of this type, myeloid-derived suppressor cells, T regulatory cells, M2 polarized macrophages, N2 polarized neutrophils, et cetera. If there is non-sterility here, then there's a whole other program that ensues, and that's really immune activation, which from a cancer standpoint is what we want. But at the end of the day, after that's taken care of, you resort back to the default, which is essentially an immune suppression, tissue regeneration, remodeling, and growth and metastases. So all by way of saying that if we go through that list and ask, are there drugs that in fact intercept that injury response, there are a host of them. So as you go through the next day and a half, what you will see initially may not make any sense, but if you start putting it in a framework, a lot of these drugs start coming together. Drugs like cimetidine, angiotensin receptor blockers, propranolol, NSAIDs, you will hear this from various speakers, and they all start making some sense. So very quickly, I've told you about the adaptive response. Here too, I'm not showing you the panoply of drugs. There are literally 2 to 3 dozen drugs that will block one or more of these pathways. And again, we'll hear a little bit about this pathway blockade a little later today. So immunotherapy, of course, the rage in cancer therapy these days, as it should be. But for immunotherapy to be successful, there are a number of things that need to happen. And these are some of them. How do you reverse tumor-induced immunosuppression? Tumor secreting cytokines that brings in those T regulatory cells, brings in MDSCs, et cetera. Cytokines such as TGF-beta, such as IL-6, IL-10, and so on. How do you initiate the immune response? How do you localize that response to the tumor? How do you maintain it? How do you create memory? So this is a cycle that has to be fulfilled for successful immunotherapy. So checkpoint inhibitors that we all know about affect the maintenance of that response by preventing exhaustion of certain T cells. There's also CTLA-4, another checkpoint inhibitor that might in fact be responsible up here in terms of and here in terms of regulating T regulatory cells. So, but additional things are needed, and this slide in kind of a messy, So for each one of these, tumor-induced immunosuppression, we can break it down. How do you neutralize the action of soluble factors? As an example, I'm not expecting you to read all this, but we can intercept at each of these levels just for this first bucket of reversing tumor-induced immunosuppression. And here are a list of drugs that will in fact hit one or more of those pathways. So this, you can argue, is theoretical and so on and so forth, but now if I show you data, that there is epidemiologic data on that somebody on a checkpoint inhibitor does better if they're on a beta blocker, that starts having some more meaning. And now if I show you preclinical data that a beta blocker plus a checkpoint inhibitor does better than a checkpoint inhibitor alone, that then takes it to the next level, which now means we ought to now put this to the test. And those are the kinds of studies that we intend to do as part of the center, and I hope many of you will join in that adventure. So just to sort of bring this to a close, how do we prioritize these? This is a scheme that we put together. Is there a fit with a scientific framework? That's what we've spent most of our time. Is there a level of evidence? What's the level of that evidence? Of course, the higher level, the better. What's the magnitude of what we might achieve? The more quantum jump there is, the more exciting things become. What is anticipated, manageable, and non-overlapping in terms of toxicities? Is there a biomarker for selecting whom we should put on a trial or for predicting who's going to respond early on in the game? And of course, cost. So this is another challenge. This was the second challenge I mentioned. It is not so straightforward as one might think. There are a lot of competing entities here. There's competition with pharma-sponsored trials. There's a general disbelief. There's a lack of awareness of what I'm talking about. And some would argue some of the preclinical and limited clinical data is just not strong enough to warrant a further study at that point. And in some cases, the mechanism of cancer action, which may be different from the mechanism which is generally thought of for that drug, has not been optimized and perhaps needs to be. So these are all the kinds of things, the kind of questions that come up when we approach folks and say, hey, is this something you might be interested in doing? And of course, the vexing issue of funding. And that's why I think a national debate about this is what's needed. How do we get the government or governments involved? How do we get philanthropy involved? How do we get foundations involved? The for-profit sector may also participate. They may want to modify the drug, which is, again, a different avenue, but at least it will move forward. They may want, for reputational reasons, to be involved. They may want to be involved if one of the cheap drugs added to an expensive one that they have a patent in makes that drug more efficacious. So we shouldn't count the for-profit sector out. And then, of course, there are opportunities in the non-for-profit areas, as we've mentioned. So why now? There's a lot happening in the world. The omics revolution is really the key to what's been going on the last few years, because we're identifying pathways from very disparate organisms, very disparate diseases that are cassettes, like the injury response, that we can bring to bear on the problems we're interested in. There's large data, which allows us to do some of the epidemiologic studies. There are some fancy technologies. And if you look at the drugs, in fact, in drug repurposing, here they are. So there's a lot of interest here, which is kind of nice. You always want to make the case. You learn from some of the folks at Harvard Business School, why now? Why is this an opportune time? So I'm trying to make the case for you. And we shouldn't ignore patients. This is something we'll talk about more at the end. Is there a way to do this in a manner that's quite radical rather than the classical studies we're talking about, typically in academic medical centers? Is there a way to distribute this into the community? And that could be a very interesting sweet spot that we'll talk about at the very, very end. So with that, let me just say, you have a pretty good idea of the way we're going to proceed. Where are the opportunities? Focus on cancer. Session 3 will focus on 2 topics, neurologic and psychiatric disease. And then issues around clinical trial design and funding to try to wrap this up. So thanks to lots of different folks I've mentioned already as I've gone through. Thanks particularly to Emory leadership. Dr. Lewin mentioned not only his support, but the support at all levels, the support of the Woodruff Foundation that got us off the ground, and more recently the support from Dr. Chan, who's sitting right here, from the Morningside Foundation. There's also a fund that we applied for, which we successfully got, called the Subvention Fund, and many of my colleagues are sitting right here in the front row and throughout that have helped enormously in getting all of this together. At the end of the day, it's about patients, and so thank you so much for coming, and let's have a good time. Thank you.